All right, picking up where we left off, we said there's a photosystem 2 and a photosystem 1. I'm going to give you an overview of how this works out. Again, you do not need to know any of these details, just simply so you've at least heard it once in your life. So we have photosystem 1, it has all these accessory pigments, and we also have the central chlorophyll molecules. The light shines in, it gets absorbed and it excites electrons. Those collapse back down and release more light. And that all focuses the light into this inner central chlorophyll duo here, because there's two pair in here. That will excite an electron, which can then get passed onto a primary electron acceptor, something analogous to NAD, or something in a protein. All right, so we need to replace the electron from here. We do that by splitting water. So we take electrons from water that is going to leave molecular oxygen, which is then going to diffuse out. All the oxygen that is in our atmosphere is ultimately from this process. Otherwise, it would be attached onto other things. All right, now once this happens and we have an excited electron, this is equivalent to an excited electron off of food. It will then get dumped onto an electron transport chain. That electron transport chain will pass electrons through a bunch of complexes, which will pump protons, in this circumstance, inside of the thylakoid space. So inside of the thylakoid, it will pump the protons into it. That produces a gradient, which can then diffuse back through an ATP synthase, which can generate the ATP. This is essentially the same ATP synthase that we see in mitochondria. Now, what then happens is that electron gets dumped onto photosystem 1. In photosystem 1, you have an analogous type of system, but light will shine onto it, and you will re-energize that electron. That will now go up into a second primary electron acceptor, which will then pass it through another mini electron transport chain, which will end up producing NADPH, which is simply a reduced form of NAD that also has a phosphate group attached. When we need electrons for biosynthesis, we usually use the phosphorylated version that helps us be able to keep a balance by choosing whether or not we phosphorylate. We, as humans, use NADPH as well. So this isn't exclusive to plants. The light system is exclusive to the plants. All right, so here at the end of the cycle, we are essentially left with two things. We've generated ATP, and we've also generated NADPH. So to put this in more of a cartoon form, here we have a low energy electron. Photons energize it, it flies up, and is then passed through a water wheel type thing, which will generate ATP. Again, those electrons are transporting protons, forms a gradient. Those protons run through ATP synthase, then falls down and gets re-energized by another photon, jumps back up, and then we can store that for use later. Now, this is not the only way it can run. This is called a linear method, right? So we start off here, the electrons go from here to here. It is a linear flow. There is an alternative to that, which is called cyclic flow. Here, if we don't need to make any sugar, we just need the ATP, we can essentially take the excited electron from photosystem 1, pass it back into the top of this electron transport chain, and keep cycling it through. This would not consume oxygen, or it would not consume water, so no oxygen would be generated. And it would also stop this whole process over here. So if the plant just needs energy, it can do this. Now, if you start looking at bacteria, photosynthetic bacteria, you can find some that have photosystem 1, but not photosystem 2. And you can, in fact, find bacteria that have photosystem 2, but not 1. And you also find bacteria that have both. So it's kind of thought that photosystem 1, as part of a cyclical flow, is probably a precursor, and then the linear flow came later on. All right, so again, coming back to this comparison of mitochondria to chloroplasts, we essentially have energized electrons going into electron transport system, which pump protons. That produces a proton gradient, which then runs back through ATP synthase, and it produces ATP. In the mitochondria, we pump those protons into this inner membrane space. In chloroplasts, they pump those protons into the inner thylakoid space. Here is another look at that system. So here we have photosystem 2. It's taking water, splitting it, energizing that electron with light, and then it passes it through an electron transport chain that is going to pump protons into this inner space. Also, splitting water generates protons, right? So it's an added function there. We then pass those electrons on to photosystem 1. It gets re-energized and then produces NADPH, which can go into the Calvin cycle. All those protons are going to leach out through ATP synthase, generating ATP, which can also go to the Calvin cycle. So let's stop and look at the Calvin cycle quickly. This is simply a process that looks somewhat similar to the citric acid cycle, but utilizes CO2 as a source to produce a sugar called glyceraldehyde 3-phosphate, which we also produce. We produce this, but we get it from sugar. All right, so this is a three-phase process. So we fix the carbon in this molecule called rubisco. We then carry out a reduction, and then we regenerate the carbon dioxide acceptor. So let's take a look at that. So here we have this 1, 2, 3, 4, 5 carbon compound. We get CO2 coming in. It is generating a 1, 2, 3, 4, 5, now 6. So each carbon is adding one more on. We can then modify this into two of these molecules, utilize ATP to turn it into this, dump electrons onto it. We now produce this molecule. This molecule can either be exported to build glucose, or it can continue on to regenerate this initial starting compound. So this is a process where we take carbon dioxide, fix it, and we can bleed out some of this G3P to use elsewhere. Now, the system has a problem, and that is rubisco, the way it recognizes carbon dioxide, it can also accidentally recognize oxygen. So, when you are dealing with a scenario where you have dehydration, you have a complicated problem. So in this situation, on hot dry days, you close your stroma in order to prevent water from escaping. This is going to reduce access to CO2 and it's going to cause the O2 to build up. As I said, Rubisco can think oxygen is CO2. Remind me to show you why this is true in class. Structurally, they can look very similar. This leads to this problem called photorespiration. So in most plants, which we call C3 plants, the initial fixation of carbon dioxide ends up forming 3-phosphoglycerate. If you do this and use oxygen instead of carbon dioxide, you end up producing a 2-carbon compound, not a 3-carbon compound, because we are not adding this carbon. This means that photorespiration is going to consume oxygen and it's going to consume organic fuel and it will release CO2 without making ATP or sugar. That is a huge problem, okay? Which is why a lot of plants will not grow in the desert, okay? There are plants that have figured out a way around this, okay? There's essentially 2 different mechanisms. There's something called a C4 mechanism and then something called a CAM mechanism. So in C4 plants, what you do is you physically separate the absorption of carbon dioxide from the environment and the Calvin cycle, right? So here we have one cell that is carrying out photosynthesis, but it is not carrying out the Calvin cycle. Instead, it takes that carbon dioxide and passes it to the next cell over. And once in those next cell over, then we can conduct the Calvin cycle. This means that in the secondary cell, you can control the amount of oxygen and carbon dioxide. So what these plants are doing, these C4 plants, is physically separating or spatially separating these 2 steps, right? So we have photosynthesis occurring over here that's generating oxygen. We take CO2 and instead capture it and then release it into another cell. So it's physically separated. CAM plants have a slightly alternative mechanism. What they do is use time separation or temporal separation of the steps. So during the night, when it is cooler, you open up the stromata and allow that CO2 to come in. You then fix it into organic compounds, and then when the daytime comes, you close those stromata, and then you can release that CO2 to start coupling it with photosynthesis in the Calvin cycle. All right, so an overview of photosynthesis. Energy entering chloroplasts as sunlight gets stored as chemical energy in organic compounds. So sugar made from chloroplasts supplies chemical energy and also supplies the carbon skeleton to produce everything the cell needs. Plants can store that sugar if it wants to. And as a side product of this, molecular oxygen is produced, which we desperately need. Taking a look at this from a pictorial overview, so we have the chloroplasts here carrying out those light reactions, the dark reactions. This is the chart that I want you to know. All that is taking place in those mesophils, which is in the leaf. We have various vasculature, so we get water from the ground. We take CO2 from the air and combine that together. We can re-export that sugar to the rest of the plant.